Good morning, everyone, and welcome. Thank you for joining us today for Cellular Conversation with a focus on plants. And we are delighted to have you with us today. And this webinar brings together two exceptional research talk given by Su Yan and Marina to demonstrate how single-cell approach is reshaping our understanding for plant biology. All the way from environmental responsive to root development, also high efficient photosynthesis in leaf. And we have our 10x Genomic Associate Director of Science and Technology Advisor, Jeff, with us today as well. And just a reminder, feel free to submit your question at any time using the Q&A function, and we will aim to address them at the end of the sections. So with that, I would like to welcome our first scientific speaker, Su Yan from Australia National University. So Su Yan will be presenting how cell type specific analysis can uncover the communication networks with chloroplasts that underpin efficient and stress resilient C4 synthesis. So Su Yan, and this is all over to you.
Thank you so much, Namchi. Let me try to get my slides up first. I believe that is working now.
Yep, so working.
Yep, awesome.
Okay.
All right, just a quick thanks to the organizers for inviting me here to speak today. And good morning, everyone. I'm Su Yan. So I'm, like Limchi said, a final year PhD candidate with the Chan Group at the Australian National University. And today I wanted to talk about some of the work that I've been doing in my PhD on unraveling the cell-specific context to stress signaling in plants that perform C4 photosynthesis. So we're all aware that climate change can negatively affect agricultural productivity, and this will lead to significant yield and profit losses. And so if we look at a local report in Australia, where we see the effects of heat stress on the wheat growing regions in Australia, what we expect is that when heat is encountered during developmental periods that are critical, such as enthesis, we would see a 20% loss in yields locally. And one way to combat this is to use what nature has developed, and that is C4 photosynthesis. So C4 plants are inherently heat tolerant. They photosynthesize at higher temperatures in the blue line shown here compared to C3 plants in the green line shown here. C4 plants are also adapted for hot, dry climates, and they have increased water use efficiency. But to harness these C4 traits for C3 crops, we need to understand what drives their resilience in the first place. And so most of what we know about how plants sense and signal environmental stress so far comes from what we've learned from Arabidopsis, which is the model for C3 photosynthesis. And using that, we've established chloroplasts as the sensors of environmental stress. So in general, what that means is that under abiotic stress, or when the stress is perceived by the chloroplasts, reactive oxygen species, or ROS, are accumulated through the photosynthetic electron transport at photosystems 1 and 2. And this ROS alters the plastid redox state, which causes damage to photosynthetic proteins and enzymes. But this also produces chloroplast signals, or retrograde signals, that can be transferred to the nucleus, where they alter gene expression towards acclimation. And this could include the induction of repair proteins as well as ROS scavengers. Two of the well-known and well-studied retrograde signaling pathways in Arabidopsis are the singlet oxygen pathway derived from photosystem 2, and that is mediated through beta-cyclocitrol and the protein MBS1. The other pathway, induced by hydrogen peroxide, or H2O2, is derived from photosystem 1, and that occurs through the cell-1 PAP pathway, with PAP being a byproduct of secondary sulfur metabolism. But one important thing to note here is that these studies were done in C3, where all the photosynthetic reactions occur within one cell type. And these studies were also done using bulk tissue analyses. And so with bulk tissue, you can mask a lot of the information from specialized cell types and specialized chloroplasts. And so in contrast, with plants that perform C4 photosynthesis, such as maize and our lab model here, Ceteria viridis, C4 plants have their photosynthetic reactions split across two specialized cells called the mesophyll cell and the bundle sheath cell. In the mesophyll chloroplast, we have the presence of both photosystems 1 and 2, whereas in the bundle sheath chloroplast, they are depleted in photosystem 2, but are enriched in photosystem 1, and they also house the Calvin cycle. Now, due to this specialization, we're actually unsure what the chloroplast signals would be and what pathways would be at play in these two cell types. And given that we have Ceteria, which is such a system with specialized cells and chloroplasts, it makes a great model for studying cell-specific retrograde signaling. So we can start by setting up our hypotheses by extrapolating what we know from C3 work. And here we have our cartoon of the Ceteria leaf with the crans type anatomy, with the mesophyll cells surrounding the bundle sheath cells in the middle. From what we know in C3 work, we expect that photosystem 2 in the mesophyll cells would generate singlet oxygen and induce beta cyclocitral signaling. And that would only be in the mesophyll cells, whereas the photosystem 1 present in both cell types would then be expected to induce H2O2 signaling through the cell 1 PAP pathway, and that would be present in both mesophyll and bundle sheath cells. We also have the epidermal layers as a major cell type, and although they're not necessarily photosynthetic, recent publications have suggested that they may have a stress sensing role. But for today's talk, I'll focus mostly on the H2O2 derived from photosystem 1 and the differences between the mesophyll and bundle sheath cells. And to do that, I'd be using bulk level transcriptomes of these cell type enriched fractions. So how can we isolate cell type enriched fractions for Ceteria? Well, I take my whole plant, treat it to combined highlight and heat stress, and then sample at a control time point as well as after 30 and 60 minutes of stress. I collect different cell type enriched fractions, in this case, the mesophyll sap, by literally rolling the leaf with a mini rolling pin. And then to collect the bundle sheath strands, I just blend the leaves. And because Ceteria leaves are so tough, whatever's left after blending are the strands. And one point to note here is that these strands, although they're enriched in bundle sheath cells, they are still a mixture of different cell types. So they also contain the vasculature, where you have your companion cells and your phloem. With these fractions, I extract their RNA and then convert that into cDNA libraries, sequence that on an Illumina platform, and then look at the differentially expressed genes. Now, if we take a closer look at the cell PAP pathway in C3 Arabidopsis, what we see is that under normal conditions, cell 1 degrades PAP into AMP and inorganic phosphate. But when stress is perceived, H2O2 is produced at photosystem 1, and this inactivates cell 1 through disulfide bonding, which allows PAP to accumulate, and it freely moves into the cytosol and into the nucleus, where it alters gene expression and upregulates stuff like APXs. If we translate this into Ceteria, we would expect both the mesophyll and bundle sheath cells to respond. However, when we look at the cell type enriched transcriptomes, that is not the case. So in this dot plot here, the colors correspond to gene expression, up or down regulated, and the size corresponds to their significance. And if we look at all of these PAP-inducible genes, which are responsible for ROS detoxification and homeostasis, we see that they are very much enriched in the mesophyll sap and not so much in the bundle sheath strands. And we do see some expression in the strands, but I'll come back to that later. So given that this gene induction is absent in the strands, does that mean it's missing this H2O2 accumulation, or is it missing this PAP signal? So one thing we can do is check for PAP, and we can do that through the HPLC, which is what we've done here. We've looked at PAP accumulation in the mesophyll sap, and we can see that it does accumulate based on the extent of stress. However, in the strands, we don't see any PAP at all. And that raises the question of, is it a detection issue or is it a biosynthetic issue? And given that PAP is involved in sulfur metabolism, we can look at the genes involved in primary sulfur metabolism, such as all the way from the sulfate transporters down to the production of glutathione. And what we see is most of these genes are primed in the bundle sheath strands, as shown here and here in the red. and not so much in the mesophyll sap. Now, if we look at the secondary branch of sulfur metabolism, which is responsible for PAP production, we see that a lot of those sulfotransferases or SOTs are upregulated in the sap. And we think that it's this one specific SOT that's driving most of the PAP accumulation that we've observed in the mesophyll sap. Now, so far, we've looked at bulk level transcriptomes of enriched fractions. And again, these are still mixed cell types. We're also missing the epidermal cells here. And so we can do better. We can take a step further to tease apart the cell-specific responses using single-cell transcriptomics. And to do single-cell transcriptomics, we need protoplasts from intact leaves. Unfortunately for me, when I started my PhD, there was no protocol existing for Soteria, and so I had the fun job of protocol development. And there were a few things that I had to optimize for, and one of them was a transcriptional inhibitor. Why this is important is because I'm working with abiotic stresses such as highlight and heat, I wouldn't want the wounding responses that would be induced from protoplasting to overshadow those. And so adding in a transcriptional inhibitor after harvesting would freeze the transcriptional state and keep the true highlight and heat stress responses intact. A few other things that I optimized for were the composition of the enzymes used, as well as the starting samples to be able to dissociate different cell types out. And I also looked at purification methods to remove any background RNA that would be contaminating my samples. So using this setup, where I heat and highlights treated my whole plants and then frozen the transcriptional states, enzymatically digested their protoplasts, and then fed those into the single cell platform. The data that I get out, I filter, I integrate, and I look at cell-specific tags. And so what does that single cell data look like? It is essentially a map of colorful dots, where each of these dots represent the transcriptomes of one single cell that has been captured. And the closer they are together, the more similar their transcriptional profiles are. And so in this data set, I've captured the profiles of about 23,000 cells under both treatments. And because they've clustered together, I can start to assign their identities based on the marker genes they're expressing. So for example, in the middle here, we have our cluster of mesophyll cells, and that is because they're expressing the light harvesting complexes associated with photosystem 2. And up here, we've got our bundle sheath cell cluster because they're expressing rubisco and malic enzyme. I've also managed to capture the cells associated with the epidermis here in the guard cells, as well as here in the pavement cells. And there's also this small cluster of vasculature cells present. Now, what can this single cell data tell us that the enriched bulk transcriptomes can't? So with the single cell data, we can start to ask interesting biological questions, and I'll demonstrate that using 2 case studies. So one, I'll be looking at the spatial component of the highlight and heat stress response using the epidermis as an example. And we can also use this data set to further tease apart the true cell-specific responses from the enriched responses. And with that, I'll be looking at the mesophyll versus the bundle sheath cells specifically, linking their differences to cell type specialization and Ross homeostasis, as well as using the single cell data set to pull out candidate genes for improving stress resilience. So if we start with the epidermal cells, so the pavement cells highlighted here in the green box, under control down here and under stress up here, what we see is a specific upregulation of genes associated with phenolic secondary metabolites induced in the pavement cells under stress. And most of these genes highlighted here are associated with the flavonoid biosynthetic pathways, as well as the isoflavonoid and anthocyanin pathways. And so the end products of these biosynthetic pathways are anthocyanins, so they can act as Ross scavengers, and you also get UV absorbers. We can try to link this gene expression with the metabolome, and one way to do that is to use mass spec imaging, where we take a cross-section of our leaf to get its chemical snapshot. And here I've used sucrose as a positive control first. So in this case, we see sucrose, or the signal for sucrose in the blue, localized to the mesophyll cells and the vascular tissue. You don't see the epidermis lit up, even though it is still present, because sucrose is not present in the epidermis usually. And so when we start to look at our intermediates of the flavonoid biosynthetic pathways, such as naringenin and genistein, what we see is signal associated with the upper epidermis. So from this, we can say that when stress is perceived, such as when a plant sees highlight stress, generally it is the upper epidermis that senses that stress first. And so it kind of makes sense to have these UV absorbers being produced in that cell layer, sort of like, I guess, plant sunscreen. And so if we switch tactics now to then look at teasing apart the cell-specific responses in mesophyll and bundle sheath cells. So here we have a schematic of the mesophyll chloroplast and the bundle sheath chloroplast with what we expect to happen with H2O2 being produced by photosystem 1 in both cases, and then the ascorbate peroxidases and the ROS homeostasis genes being upregulated to deal with that accumulated H2O2. But what we saw from the enriched fractions is that that wasn't the case. So we only saw upregulation of these ROS genes in the mesophyll sap and not so much in the bundle sheath strands. If we look at the single cell data, we do see that trend being maintained, where under stress in the blue violin plots down here, we see the upregulation of these genes in the mesophyll cell clusters. But when we start to look at the cells that make up the bundle sheath strands, we can split them into the bundle sheath cells themselves, as well as the vasculature cells. And what we see is that gene expression we saw in the enriched fractions before is actually localized to the vasculature rather than to the bundle sheath cells themselves. And so this lack of APX expression in the bundle sheath cells, does this then translate to differential patterns of H2O2 accumulation in live tissue? And we can check that by staining for H2O2 in whole leaves using the fluorescent ROS dye called DCF. And under control conditions, we don't see much signal being present. However, after 30 minutes of stress, we see H2O2 signal associated with pretty much all cell types and in both the cytosol and chloroplasts of these cells. What's interesting is that after 60 minutes, most of that mesophilic H2O2 has dissipated, leaving only the H2O2 associated with the bundle sheath cells behind. And so that then raises the question, why are bundle sheath cells not upregulating their APXs to deal with this elevated H2O2? Why do they have that elevated H2O2 in the first place? And what happens if we try to mimic or force cell on-tap signaling in these cells by forcing APXs to be expressed in that cell type? And that was exactly what I did, where I generated different mutant lines that were overexpressing APXs either in all cell types or specifically in the bundle sheath cells with a bundle sheath promoter. I then treated those plants to a detached leaf stress assay, measured their ROS levels, and then measured their FEFM for photoinhibition. And what I saw was that here again, we're looking at H2O2 staining, where the yellow signal corresponds to H2O2 with the magenta signal corresponding to chlorophyll, and the blue arrows indicate your mesophyll cells or where they would be localized, and the orange arrows would indicate where the bundle sheath cells are. And under this stress, both the wild type and the negative control show no difference. So again, they show upregulation or accumulation of H2O2 pretty much in all the cell types and in both the cytosol and the chloroplast. When we look at the plants that were overexpressing APXs in all the cell types, what we expected was a reduction in H2O2 levels. And yes, that is what we see, a reduction in H2O2 in both cell types. And all the signal that's remaining is associated with the chloroplast, which makes sense because we overexpressed a cytosolic APX. What's interesting though, is when we look at the lines expressing bundle sheath specific APX, they actually look quite similar to the lines expressing APXs in all the cells in terms of the staining pattern. But does this reduction in ROS and does this staining pattern then translate to any sort of stress resilience that we can measure? And to do that, I looked at their FEFM, which measures photoinhibition or the health of PS2, mainly in the mesophyll cells. And what we saw is that in the lines overexpressing APXs, compared to the wild type, they both have less photoinhibition. But what's interesting is that the lines that express APXs only in the bundle sheet cells have less photoinhibition than the lines that are overexpressing APXs in all the cell types, which was quite interesting, quite surprising. And to summarize all these findings that are presented here today into sort of a preliminary model, what we saw was that different cell types actually use different types of chloroplast signals and stress responses when they encounter highlight and heat. And so when highlight and heat is present in the pavement cells, we saw UV absorbers being upregulated and being synthesized. And that's for, I guess, UV protection against highlight. In the mesophyll and bundle sheet cells, we saw an accumulation of H2O2. And what's interesting was in the mesophyll cells, because PAP was present, it kickstarted cell PAP signaling, and that resulted in the expression of the APXs, which dealt with that excess H2O2. But because the bundle sheet cells were not able to express APXs, they instead accumulated H2O2. And so this shows us that C4 is actually a great model for investigating retrograde signaling because of its specialized cell types and chloroplasts. And hopefully through this presentation, I've shown some pros and cons that are associated with using enriched fractions versus single cells when looking at transcriptomics, as well as introduced some of the spatial responses that occur in plants like Ceteria. And beyond this, we can start to question or probe if there's any sort of intercellular signaling going on between these mesophyll and bundle sheet cells, given that they're able to sort of partition that ROS accumulation between the two cell types. And from this, we've sort of started to provide some novel strategies for enhancing stress resilience and crop productivity by looking more at cell-specific gene manipulation rather than whole plant gene manipulation. And just to sum up, I'd like to quickly acknowledge my supervisory panel for all their support, as well as the group here at ANU and some of our international collaborators at Ghent and University of Copenhagen. And thank you all for your attention.
Thank you. Thank you, Shuyan, for the excellent presentation. Really impressive to see how single cell approach fit in with other technology like mass spectrometer and metabolomic analysis. So we will keep the question to the end. So now our second speaker is Marina Oliver from University of Western Australia. Marina going to show us how plant roots respond to environmental signal through distinct cell lineage pathway and try to review different mechanisms plant use to adapt to growth and development under changing conditions. So before handing over to Marina, I would also like to extend my special thank you to Marina, who joined us from Western Australia, which is 3 hours behind East Coast. So really appreciate you making the time to be with us today. And Marina, I will hand it over to you.
Thank you, Lim-Chi. So first, thanks for inviting me today to present my work. So I'm Marina. I'm a postdoc at the University of Western Australia, as Lim-Chi said. And today I'm going to talk about how we explored cell identities in the root using single cell transcriptomics and how it helped us uncover a new cellular state that is important for environmental responses. Whoops. Okay, so imagine most of you attending the webinar today must be plant biologists and must be familiar with the Arabidopsis root. But if some of you are not, just briefly introduce my biological system. So this is a schematic, both a longitudinal and a cross-section of the Arabidopsis root that is composed of different cell types that you see here in different colors. And as you can see, these cell types are really well organized in concentric layers. And what is really nice with this system is that roots are continuously growing throughout the plant's life, right? So during all its lifetime, the plant is going to be continuously producing all these different cell types. And this is because there is a small pool of stem cells here that you see in white at the root tip that is going to be continuously producing new daughter cells. And as new cells are being produced next to the tip, the older cells will move along this axis and differentiate. So if we focus here on this pink cell type in the middle of the root, we're going to have a gradient that goes from an undifferentiated stem cell to a fully differentiated, elongated functional cell. So this system is the perfect system to do single cell on, because when we take each cell individually, not only they give information about the spatial arrangement of cells, but each of them are also a snapshot in a dynamic process that is cell differentiation. So they each hold, let's say, a temporal information about a biological process. So we thought this was a great idea, a great model to do single-cell RNA-seq on. And what we expect to do is not only to identify the different cell type and their transcriptomic signature, but also to identify all the different intermediate in this differentiation process for each of the lineages of the root. So this is what we did, and we obtained an atlas of the Arabidopsis root tip that is composed of a bit more than 87,000 cells from 9 different samples. And the first step we did is to define cell clusters and to find genes that are specific to each of those clusters, so we could assign a cell identity to each of them. But instead of just looking for genes that are enriched in clusters and intersecting them with known genes, known cell type specific genes, we decided to develop an experimental validation of our cell type identification. So instead of just intersecting gene lists, we wanted to really look in vivo at the expression of all these cluster specific genes that I showed before. So to do so, we developed a whole mount in situ hybridization protocol to visualize gene expression directly in 3D in clear tissue without the need of cutting the tissues in sections. And we combine this whole mount in situ hybridization method with the HCR method that you might have heard of for transcript detection, which uses basically a system of split probes and an amplification step with a hybridization chain reaction of fragments that are coupled to fluorophore. And this system basically drastically reduces background in situ hybridization. So we applied that to all the different cluster-specific genes, and you can see in these movies that we obtain very specific gene expression here at the center of the root in clear tissues directly in 3D. So we used that to assign an identity to all the different clusters that we had. I don't know if you can see there you go. Identified in the atlas. And the good news is that we could identify all the different cell types in the root that I've put here. So the first thing we can see is that we have groups of cells, and these groups of cells basically correspond to the different cell lineages of the root. So this confirms that the lineage or cell type information is the strongest discriminant in this data set, right? The first thing that separates the cells in the atlas is the lineage they belong to. But now that we have each of the lineages, what we want to do is be able to order the cells within each of these lineages to recapitulate this developmental progression that I talked about in the introduction. So how do we look at developmental progression within each of these groups? So we're going to treat them each individually, independently, and try to organize the cells within them. So first, I'm going to introduce a little bit the trajectory inference method that we used for organizing the cells that is called partition-based graph abstraction, or PAGA. So it's composed of 2 different steps. The first step is to subdivide the lineage into small clusters and then compute the connectivity, which basically it's a proxy for similarity, between them. This will give us the topology of the trajectory, or the relationship between the cells, the shape of the trajectory. but we have the shape and we don't have an orientation. We could imagine that the trajectory starts at the end of any of the branches, for instance. If it starts here and then it develops and then splits into two different branches. So we need to be able to put an orientation to this trajectory. So to do so, we use the second step, that is diffusion pseudotime. So basically, we need to identify in the data set which cell is going to be considered as T0, based just on biological knowledge of the system. In our case, for instance, we can look at genes that we know are expressed or enriched close to the tip, close to the stem cells, and just identify the genes that expresses these genes at the highest level and identify this cell as T0. So what the software would do is, based on this assigned T0 cell, it will look at gradual variation of gene expression in the data and assign a pseudotime to each of the cells. This pseudotime is simply going to be a zero number between 0 and 1, 0 being the closest to this initial state, and the cells that will be assigned 1 will be the most different from this initial state. And by doing so, we're going to just add an orientation to the trajectory and then obtain the lineage trajectories. For instance, here we put T0 in the middle. So instead of having to starting here and dividing into two branches, we know that we start here and we have 3 different developmental branches. So this is just to explain the methodology. And what we've done is that we've applied that methodology to each of the cell lineages in the root independently. So here you can see the different groups subdivided into smaller subclusters that are of different colors. And here the diffusion pseudotime analysis we obtained. And by combining these two things, we can obtain an abstracted graph. So in this graph, each dot is a subcluster. The lines here connect the subclusters of the same lineage. And thanks to the pseudotime information, we can put an orientation to those branches that are represented here by arrows. So this looks very complicated. So I'm just going to focus on one lineage to show you the result we obtained. So if we focus here on this pink group of cells, this is the endodermis. So this is one lineage. We could expect T0 to be at either end of this group of cells and to have a linear trajectory of differentiation that goes from one end of this group of cells to the other. But what we saw is that actually T0 falls somewhere here in the middle of this group of cells. And we can identify subclusters on both sides that are connected, which means that we don't have one branch, one linear trajectory, but instead we have 2 branches, right? So T0 is here in the middle, and we connect these clusters on one side and the clusters on the other side. And the pseudotime progression goes in this direction and in this direction, as you can see here. Pseudotime is 0 here, 1 on this end, and 1 on the other end. And this applies to all lineages. I gave you the example here of the endodermis, but if we looked at the xylem pole pericycle, we see exactly the same thing, 2 branches. Same for the phloem pole pericycle, so on and so forth. So believe me, we see that for all of them. For now, we arbitrarily call them branch 1 and branch 2 because we don't know much about these. So what we obtained from the single cell trajectory is quite different from what we would expect from textbooks. We would expect to have only one linear trajectory, but instead it seems that we have 2 different possible trajectories for each of the lineages. So what are they? What differentiates branch 1 and branch 2? So we could have done just per lineage a differential gene expression between branch 1 and branch 2, but instead we chose to do things a little bit differently. We looked at gene expression dynamics across the entire data set, and we looked for modules of co-expressed genes. So we hope to find co-expressed genes that are going to be specific to one cell type or to one branch, for instance. But by using the entire data set, we also expect to see commonalities. For instance, we might have some genes that are going to be specific to multiple cell types or multiple branches. And so by doing that, rather than looking at each lineage independently, we can also find these commonalities. So we detected in total 25 co-expression modules. Some of them are specific to one cell type, for instance, here, the root cap, or here in the epidermis, while some others are specific to multiple cell types. For instance, these modules here are specific to the stele. So the stele is all the vasculature of the root, which is comprised of multiple cell types, the percambion, the xylem, the phloem. So we can see here that these genes are expressed in multiple cell types. We also have some ubiquitous genes that are expressed in basically the entire root except the root cap. But what caught our attention is that we had two modules, module 20 and 21, that were found across all cell type, but were specific to one of the two branches. You can see here on the UMAP. So remember, each of these groups is a cell type, but we do have branch 1 on one side and branch 2 on the other side. And you can see that both those modules are enriched in branch 1, but barely detected in branch 2. And this is not just a few genes, because these two modules are the third and second most biggest module in terms of number of genes. So it's quite a lot of genes actually that are branch specific. And this is confirmed if we look just on the UMAP at the number of genes per cell or the number of UMI per cell. We can see that branch 1 here in multiple cell types consistently have more genes and more UMI. This means that it's as if a subset of the cells of all cell types have superimposed on their cell type identity a strong transcriptional activity, right? And this is what differentiates the cells that we labeled as branch 1 of the cells that we labeled as branch 2. And this is happening regardless of their developmental origin. The root is going to have a subset of the cells that are going to have that, and is going to affect all the different cell types, which is really something we didn't expect when we started this project. So obviously, the first thing that comes to mind when we find something we didn't expect, and we see that it involves having different gene and UMI content, is to think that this is an artifact. May it be experimental or computational. We have low quality cells, or we have something going on that makes some cells have fewer genes and fewer UMIs, but this is not a biological reality. So a lot of the work we've done is trying to rule out many possible artifacts that could have caused that. And I'm going to introduce to you just a few of the ones we've checked without going too much into details. But just to give you a few examples. So the first thing we thought of is in just different cells of different cell types at different cell cycle stages. So we've assigned a cell cycle stage to each of the cell, and we couldn't really see the branch 1 and branch 2 separating according to cell cycle stage. So we knew it wasn't that. The second thing we thought of is just protoplasting. So to do single-cell RNA-seq, we needed to dissociate the tissue into protoplast, which means digesting the cell wall. And this process will trigger changes in gene expression. So what we've done is we've looked at changes in gene expression between dissociated root and undissociated root. And here you have a diagram showing the log fold change of the genes grouped by the co-expression modules that I introduced earlier. And here we have module 20 and 21, which are the branch-specific modules. And we see that the genes in those modules are not specifically overexpressed or upregulated, or downregulated during the tissue dissociation. So we cannot explain the expression of these groups of genes simply by protoplast generation. The other thing we thought of is, okay, we have branch 2, which are low UMI cells of the same cell type of branch 1. So couldn't they just be damaged cells? If it was the case, if there were damaged cells that were leaking RNA, we would somehow expect the genes that are detected only in branch 1 and not in branch 2 or low quality cells to be the low expressed genes. Why? Right? So the lower the expression the more chances we have of not detecting them in the damaged cells. So we compare the expression level of the genes that are expressed both in branch 1 and branch 2, and the genes that are expressed only in branch 1. And we could see that they basically span the same expression range. So we know that the genes that are detected only in branch 1 are not just low expressed genes that wouldn't be detected in damaged cells. Also, when we think of damaged cells, we think basically of nuclei, right? So are we looking at cell versus nuclei in our data set? To test this hypothesis, what we've done is that we've used single nuclei RNA-seq data from the Arabidopsis root tip that was already published. And we thought, okay, if that's the case, then we shouldn't see both branches in single nuclei RNA-seq. So this is a UMAP showing a projection of nuclei from a published data set onto our reference atlas. And what we can see is that we see nuclei that are assigned at branch 1 and branch 2 in all cell type of the data set. And what's interesting is that in the nuclei data set, we also see a difference in UMI counts between branch 1 and branch 2. And if we look at the genes, oh, it's really small, I don't know if you can see, of module 20 and module 21, we see that they are also enriched in branch 1. So we detect the two branches in nuclei. They also have a difference in UMI content. And this is also explained by the same genes as the ones we detect in our atlas. We also checked other single cell data sets that are already published. So this is the biggest single cell root tip data set published to date, where we can see the different cell types. Here, they are actually connected together on the UMAP close to the tip. So this is the trajectory of differentiation of one cell type, another cell type. And you can directly see on their UMAP that all these different cell types at the end of the trajectory seem to split into two branches. Even though it wasn't really explored in their study, we can clearly see it on the UMAP. And if we look at the UMI count, we also see that we have a difference between branch 1 and branch 2. Well, one is 1 and the other is 2. And we also see that the genes of module 20 and 21 that we identified in our study are also enriched in only one of the two branches. But ultimately, what we can do to prove that what we see is just not a computational or an experimental artifact is go back to an orthogonal validation using the whole mount HCR. So this time we looked at the transcripts of two genes that in our single cell RNA-seq is shown to be branch 1 specific. And what we expect is to see cells that express it and cells that do not express it within one cell type. So this is what we see here with the top gene. We see in the epidermal layer, we clearly have some here longitudinal section and cross section. We clearly have cells that express these genes next to cells that do not. And same here with that gene. And we can see in the inner layer here as well of the cortex, we also have that. So we have basically experimental validation that what we're looking at is real and not just an artifact. Okay, so now that we're convinced that we're looking at something biologically meaningful, we need to understand what might be the role of the cell state in the root. So the first thing we did is just to do a go-term enrichment for the genes in these two modules that are branch 1 specific. And we came up with a lot of terms that are linked to environmental responses. For instance, responses to nutrients, responses to stimulus, the circadian clock as well, transcription, because we have cells that are a lot more transcriptionally active, but also response to ABA, which is a signaling pathway, so plant hormone that is known to mediate a lot of the abiotic stress responses. So because we got this, we're going to call this state the environmentally responsive state, or ERS. And the branch cells that have this transcriptional activity will be the ERS positive cells, and the branch 2 cells, we don't have that transcriptional activity, will be the ERS negative cells. So now that we've done that, we have a hypothesis to test, which is that this cellular state that we identified is important for response to environmental stimuli. So the first thing we did is we finally had a signaling pathway that seemed to be involved in the formation of this state. So could we test that biologically? So what we did is we profiled the root tip of two different types of ABA mutants, an ABA biosynthesis mutant, so we had an ABA deficient mutant, and a quintuple mutant for ABA receptors, so an ABA insensitive mutant. And so we had 3 replicates for each of these conditions, and this is the UMAP we obtained. And we could detect, let's say here in this group, this is xylem pole pericycle, the 2 different branches, right? We still have 2 populations. But when we quantify the proportion of ERS negative cells and ERS positive cells, so this is a symmetric, this plot is a symmetric of that one, because cells can either be ERS positive or ERS negative. We could see that in both mutants, we have a strong and significant reduction of the proportion of ERS positive cells as compared to our wild type. And if we look at gene expression, and more specifically at the 25 modules we defined in the study, so here the modules have been ranked based on their average log-fold change in the mutants. So here on this side, they are upregulated. On this side, they are downregulated. The 2 modules that are most strongly downregulated in both mutants are the module 20 and 21, which are ERS specific. So the ERS is significantly reduced in ABA mutants, showing that the prediction that the GO term analysis made is probably right, that this pathway is important for the formation of this cellular state. And we know that ABA is an important mediator of drought responses in plants. And because we also got a lot of terms that were linked to water responses and nutrient responses, we decided to test this hypothesis. So what we did is we grew plants in conditions with much, much lower water content, just by simply reducing the water content in the MS medium to 50% or 20% of this of its normal content. And it has been shown in a study to trigger the same changes in gene expression as drought on soil. And when we do that, we already see in the UMAP that we have a little bit less of the 2 branches when we hit the 20% water as compared to the control. And we can quantify that. So here on this axis, we have increasing water content. So increasing drought stress from right to left. And this is a proportion of ERS negative and ERS positive cells. And what we can see is as we increase the drought stress, we also increase the proportion of cells that acquire this ERS, right? And also, and then consequently a decrease of the ERS negative cells. And we could confirm that again using the whole mount HCR. So we look at 2 genes that are ERS specific or branch 1 specific. We can see in normal water content that we have cells that express the genes and cells that don't express them. But as soon as we put the roots under a very strong drought stress, all of a sudden we significantly increase the number of cells that are going to express these genes. And this can be quantified by fluorescent measurements. So basically, the root is able to adapt the proportion of the cells that's going to acquire that new state under drought conditions. So to summarize, what we could show using single-cell RNA-seq is just to show that we have another layer of regulation of cell identity in the root. So we can think of different layers of cell identity as if they were nested Russian dolls, right? At the core, we have a very stable identity, which is the cell lineage. And that is established during development, and it is very stable. Then on top of that, we have the cell differentiation information with this gradient, which means within each of these layers, we're going to have that other type of information, which is the cell differentiated or undifferentiated? And what we show is that we have another layer of identity, which is, do the cells have the ERS or do they not have it? And what we show is that this layer of identity is very dynamic and responsive to environmental conditions. And because this is just a 10x user meeting, I think there are two important takeaway messages from this study as well, is that low UMI cells don't necessarily mean low quality cells. Sometimes they do, but sometimes they don't. So a lot of the root cell study have discarded the branch 2 cells, thinking they were low quality cells. But instead, we've shown that there is differences in transcriptional activity within one cell type. And the second thing is, I think, the importance of orthogonal validation methods to single-cell RNA-seq, because if we didn't have the HCR, whole mount HCR, we wouldn't have been able to fully convince ourselves and the reviewers that what we're looking at was real. So thank you for your attention. I'd like to acknowledge all the people that have been involved in this study, and thank you for your attention.
Thank you, Marina. Thank you for your wonderful talk. It's really nice to see how single cell can not only used to study cell type, but also cell state and cell lineage. And do we have any question from the audience? I don't see any question. I do have one for Shu Yan. So when I saw your UMAP, I saw that there's some unassigned cluster. So do you have plan for following up for those clusters?
Yes, so at the moment, we think it may be a cell state, given what Marina's talked about as well. But we also think it could be a cell type that doesn't have known markers at the moment, given that the Ceteria genome is a bit under annotated. So we were hoping to get some in situ PCRs going for this, but that didn't work out for Ceteria. So what we're hoping to do next is to generate or pull out some high expressing genes in that cluster and make reporter lines for that in Ceteria.
I see.
We'll see.
Yeah. And for Marina, I noticed there's no branch 2 specific module. Is that right?
Yeah, so what the data seems to show us is both branch 1 and branch 2 will have the cell type specific, but branch 1 will have that additional layer. That's why we call it layer, right?
Yeah.
But, and I haven't shown it here, but hopefully you'll see it once it's published.
Yeah.
The only cell type for which we could find branch 2 markers are the hair cells in the epidermis. And I've done a whole mount in situ using both branch 1 and branch 2 for the hair cells, and we can see them expressed next to each other, but in different cells. So it further confirms that what we're looking at is true. But the thing is, we don't have globally, when we look at the co-expression modules, let's say a genetic program that is specific to branch 2, right? It's rather, it's specific to the cell type, and then the branch 1 will have this information on top of the cell type activity.
I see. All right, thank you. And thank you all for joining us today. A special thanks to Shu Yan and Marina for sharing their research, and wishing you best of luck, both on writing your thesis and publishing. And thank you everyone for attending. A recording will be shared to all the registrants. And if you need more information, do reach out to us. And thank you again. Looking forward to see you in the future events.
Sorry.
Thank you.
Yeah.
There is a question in the chat.
Oh, really?
Yes.
Just come up.
Just came in last minute.
Oh, yeah. Lilian, yes. Hi, Lilian. So Lilian asked, thank you for the nice presentation. And have you ever tried to isolate nuclei instead of protoplasts? And do you have changes to the gene expression due to getting protoplasts?
So, go
Sorry,
ahead.
yeah. So I can take that first. We did consider both single nuclei and single cell. So looking at nuclei or protoplasts. For us, it came down to how much information we could get out of the system, because we're looking at stress. Most of the stress responsive transcripts are cytosolic. So if we were to capture nuclei, we would lose a lot of that information. And a lot of the stress responsive transcripts also undergo alternative splicing. Again, if we use nuclei, we would lose that information. So we opted to go with protoplasting instead. But yes, there is a big effect of protoplasting on gene expression where you get wounding. And so, which was why we went with transcriptional inhibition via chemical methods. And I think a few papers that are coming out now have started to include that in their protocol. And when single cell first started, mostly people were looking at cell identity. So wounding expression wasn't a big deal back then. But now that we're looking at treatments, that's more of an issue. So yeah.
So we haven't tried nuclei extraction, and I think there's pro and cons, right, for each method. But I think if we were to chop the tissue to get nuclei, we would lose a lot of the, the rare cell types at the beginning of the trajectory. So that might not be ideal. And this is what we see with when we project the nuclei data onto our atlas, we lose a little bit of the early processes. So what we had tried as well the transcriptional inhibitor when we started the project. But we detected that it reduced a lot the number of transcript that we could detect per cell. So what we did instead is to do just an experiment where we compare unprotoplastid versus protoplastid root, and we look at the genes that are up or down regulated, and we remove them from the features when doing the clustering, the dimension reduction, all of that. So we remove the protoplastin induced or sensitive, genes from the first step of the analysis, and we can still see the different cell types and the different branches. So that's one way to address that issue.
Cool. Thank you. Thank you for the answer. I think that is the question. All right. Cool. Thank you, Lian. All right. Thank you, everyone. See you at the next webinar.
Thank you.
Thank you, Jeff.